Good afternoon, ladies and gentlemen, colleagues, and welcome to this year's G. Burroughs MIDA lecture as part of the Wednesday afternoon lecture series. It's my great pleasure to have the opportunity to introduce our speaker today. But before I do, just to let those of you who don't know that the lectureship was established in 1968 in honor of Dr. MIDA, who is the first NIH Director of Laboratories and Clinics. And each year, this lecture is presented by an NIH intramural scientist who is recognized for outstanding contributions to biomedical research. And today, our speaker is Dr. Ronald Germain, truly one of our most creative and productive intramural scientists. Ron is the chief of the NIAID Laboratory of Systems Biology, and since Bill Paul's untimely death in September, Ron also has served as the acting chief of NIAID's Laboratory of Immunology. Ron received his MD and his PhD from Harvard in 1976. The PhD was for research with Baruch Ben-Asaraf, the recipient of the 1980 Nobel Prize in Physiology and Medicine. And Ron began his career on the faculty at Harvard Medical School, and since 1982, has been the chief of the lymphocyte biology section right here at NIAID in the Laboratory of Immunology. Over the years, Ron and his colleagues have made contributions to our understanding of the major histocompatibility complex class II molecule structure-function relationships, the cell biology of antigen processing, and the molecular basis of T-cell recognition. And most recently, his laboratory has focused on the relationship between immune tissue organization and control of adaptive immunity, which was studied by utilizing novel, dynamic, and static in situ microscopic methods that his laboratory actually helped pioneer. Ron's published 300 papers, more than 300 papers, and among his many honors is election to the National Academy of Medicine of the National Academy of Sciences. He co-founded the NIH Immunology Interest Group and Systems Biology Interest Group, and he acts as the Associate Director for the Trans-NIH Center for Human Immunology, Autoimmunity, and Inflammation. He also has been an extraordinary mentor, training more than 70 postdoctoral fellows, some of whom have gone on to become scientific leaders at top universities and medical schools around the world. His lecture today is entitled Imaging Immunity, during which he will hear about the elegant experiments that are giving us a more complete picture of the operation of the immune system. So please join me in welcoming Dr. Ron Germain.
Well, thank you very much for that extraordinarily kind introduction, and I'm also really grateful for my colleagues having nominated me for this honor. It's always good that your colleagues and I hope friends think that of you. And I want to start in a little bit of an unusual way, which is with this picture, which is typically at the end of a talk. But often what people do is to show this picture and say, these are the people who did the work. And I think that demeans these individuals. These are my colleagues that I've worked with cheek and jowl in the lab for years. They are creative individuals. They have insight into the biology, and some of them are pretty good technically. And it's their work in collaboration over the years that really has produced all the research results and the insights that I hope to be able to convey today. So this is an early picture at the beginning of our journey in imaging immunity, an intermediate set of folks, and some of the very recent people. And I will call out the individuals who contributed each of the different stories that I will tell you. I also want to thank the DIR of NIAID for all these years of support, without which I could not, and my colleagues could not, have conducted this research. So we live in a dangerous world. We're constantly bombarded with potentially pathogenic bacteria or viruses, fungi or parasites. And we also have a dangerous internal world with the possibility of malignancy developing in any of the different tissues in the body. And this raises an important question. How does the immune system protect against diverse, unpredictable disease entities at unanticipated sites in the body? And so the strategy that has been developed in evolution is to combine organized tissues that themselves are distributed throughout the body, and I'll highlight a few of those in a moment, and mobile elements. And so the fixed tissues include the primary lymphoid tissues such as the thymus gland or the bone marrow that produce important cells, as I'll mention in a moment. We have distributed lymph nodes throughout the body, specialized mucosal secondary lymphoid tissues such as pyres, patches, and we importantly have these formed elements, the combination of myeloid and lymphoid cells that are primarily produced in the bone marrow. Most of the myeloid cells exit as mature cells into the bloodstream. Some of the lymphocytes, particularly B lymphocytes, are at least semi-mature or mature when they exit. Others exit as precursors. The mature myeloid cells can rapidly, as part of the innate immune system, find inflamed endothelium at sites of tissue damage or pathogen invasion, transmigrate and mediate their effector function in that location. The precursors that I've mentioned can enter the thymus, move from the cortex to the medulla, and then back into the vascular system to circulate to reach the secondary lymphoid tissues. And there, these T cells, along with the naive B cells not shown here, can enter through high endothelial vanules into the parenchymal part of the lymph node. The lymph node itself has an organized structure. It has a paracortical T cell region and these primary B cell follicles. And yet, although those are canonical structures, each of the individual lymphocytes that's present there is constantly changing and turning over as cells circulate and move through this tissue. What are they doing there? Well, they're looking to meet up with their cognate antigen. These lymphocytes have clonally distributed receptors, and when those are engaged, these cells get activated. For T cells, the relevant ligand is this peptide MHC complex, a surface-disposed piece of the antigenic components of, say, a virus or a bacterium. And these dendritic cells might either be resident in the lymph node, or they might migrate in through the afferent lymph. In that compartment, these rare, relevant T cells might meet up with a rare, relevant dendritic cell bearing the right antigen, in which case they can become activated, divide, become effector cells, leave through the efferent lymph, move to inflamed endothelium, and like the myeloid cells I mentioned, move into the tissue to mediate effector function. And we've known these general outlines of the migratory behavior of cells in the immune system for decades. But there are finer-grained questions that arise in thinking about how this all might work. And some of those questions are, what are the real cell dynamics? How fast do lymphocytes move through a tissue or myeloid cells in a lesion site? And how do those dynamics relate to their function? Do the cells mediate effector function when they're moving or when they're static? How long do they spend engaged with each other as they're exchanging signals that we know are critical for the development of good immune responses? The second question relates to what I just showed you in the lymph node. There are organized substructures. These are not just bags of cells. And the question is, why are those tissues organized in that manner? And are there finer-grained aspects of this tissue anatomy that we haven't recognized in the past that are connected to the functioning, the efficient functioning, and I'll stress this issue of efficiency, the efficient functioning of the immune system? And then, of course, at a finer grain level, what are the molecular signals that guide these movements and positioning, control interactions, and regulate activity in a complex tissue environment, not in a tissue culture flask? And so the approach we and others developed to begin to approach these questions about 15 years ago was using multiphoton intravital microscopy. And so in outline form, we expose the tissue of interest, and I'm going to tell you what some of the problems are with this method, in contrast to most people promoting the methods they use in the lab. And the first thing is we have to do surgery except if we look at the skin. And that means we're introducing potential inflammatory signals, and we have to be very careful and run controls to know that when we say we're looking at the steady state properties of a tissue, that we've measured whether there's vascular leak. We measure whether there are neutrophils that show up where they don't belong to see if we've induced a level of inflammation that would distort our understanding of basic physiology. Once we've exposed the tissue, we can take a pulse laser and scan it in an XY direction to create a plane of imaging information, and then focus up and down to get a stack of information. And then we repeat this over time to get 4-dimensional data sets that we play back only in 2 dimensions because NIH cannot afford holographic projectors. So we're looking here at T lymphocytes moving in the paracortical region in a lymph node. And so the second point is you see green cells moving in an inky black background, and they look like they can go anywhere they want. But the reality is they're in this environment. They're always touching another cell that's not fluorescent, a nerve cell, a stromal cell, something else. And so you should never trust an imager who draws conclusions based on what he or she does not see. And since we only make a limited number of components fluorescent, we're always not seeing a lot of things, and we have to be very careful how we do our interpretation. And I'll show you some of the tricks we use to try to minimize this as a problem in our imaging experiments. So the immune system is composed of different layers, and the 3 essential layers are the barrier functions, the skin and mucous membrane, that physically keep the pathogens out. If that's breached, we can get rapid, generic, if you will, innate immune responses, and then eventually the development of the type of adaptive immunity we're most familiar with from resistance post-infection or from vaccines. And so the talk is organized in this flow of the immune function. So let's talk about a barrier breach. This is work we did years ago with David Sachs in the Laboratory of Parasitic Diseases, and we're looking at a sandfly successfully having a blood meal in the skin. And if we look in a mouse that has a sandfly bite, we're looking in the ear skin, we're looking flat on this thin ear. There's a blood vessel running here that you'll see some tracer in. This is the site of bite damage. Green cells are genetically EGFP positive or GFP fluorescent protein positive neutrophils, and these small red spots are genetically labeled Leishmania organisms that have been deposited by the sandfly bite. And when we look at this, we get a lot of information. For people who study chemokines and chemokine receptors and cell migration, what we see is how precise guidance is in vivo. No neutrophil leaves the wrong side of the blood vessel, even though it's only 1 or 2 cell diameters thick relative to where the damage or the infection is. We see these long strands. That is indicative of the difficulty of cells that have upregulated integrin affinity. That's part of their trans endothelial migration, releasing themselves from that tight adhesion. There's very directed migration and then swarming at the site of damage. And this is all really quite informative, but it's also only looking at it essentially in 2 physical dimensions. If we turn everything on the side, and this blue is a free channel we get from the collagen in the dermis, black is not empty space, but the epidermis, we see these green cells jumping into particular places here. And what are they doing? They're actually filling in the barrier breach. We don't want to have that barrier exposed so that organisms can continue to invade into the tissue, or even worse, into the circulation. And so these neutrophils are carrying out very complex behavior in 3 dimensions plus time in order to mediate host defense. And so we understand very well this paradigm of select enrolling, adhesion, and integrin-mediated transmigration of getting neutrophils into a tissue site. But what really controls their activity of the type that I just showed you in very complex 3-dimensional environments with extracellular matrix components, DAMPs from tissue damage, PAMPs from pathogen invasion, many different cell types such as tissue-resident macrophages, plus mesenchymal cells of varying types. And so over the last few years, Tim Lammerman really developed an extraordinary system for exploring this and did some actually terrific work. And we did this in collaboration with Carol Parent, who had done some in vitro work that's highly relevant for the model that emerged from the in vivo imaging. And so what Tim set up was a sterile injury model. We used the laser to create a very small damage of 5 to 35 or 40 microns. We now no longer have a barrier breach, and we know this is sterile. We're not moving commensal PAMPs into the tissue by sticking a needle through something. We can make sure if we use a blood tracer, we don't nick a vessel, and we therefore don't have the involvement of platelets, which is true in other models of so-called sterile injury. And we know precisely when the lesion was made, so we can do kinetic analysis. So if we look in an animal now where the neutrophils, instead of being green, are red, and the green cells are monocytes, we see that swarming behavior that I mentioned previously, and over the 2 hours of this movie, this gradual accumulation of monocytes as a ring around the central neutrophil swarm. If we deplete the neutrophils, the monocytes do not find the central lesion. So they are being recruited by the neutrophils indirectly. The problem with looking at these endogenously labeled systems is we want to look at behavior in the tissue where we think that integrins and chemokine receptors may play a role, and they might overlap with those that we need for transmigration and exit from the vasculature, in which case we'd have no cells to examine in the tissue. And so what Tim worked out, which was rather difficult to do but works very nicely, is adoptive transfer of isolated dye-labeled neutrophils, which after a few hours will move away from the injection site, and then we can create the lesion and then look at their behavior in response to that lesion in the tissue. This avoids the problem of not getting the cells out. It actually also works with human neutrophils, which will not come out of a mouse circulation because there's no IL-8. They move a little more slowly, but they respond, which means you can look at genetically variant neutrophils, or you can do drug treatment in the context of an intact animal rather than an endothelial layer in culture. And it's much cheaper and faster because you don't have to breed the animals to the reporter. You can take bone marrow from any knockout mouse, dye label it, and do the experiment quickly. And so it looks just like the endogenous. If I didn't tell you that this were the transferred cells, you wouldn't be able to tell it from what I showed you before. And by all measurements that we can make quantitatively, there's no difference in the behavior. But while looking at this, we also were looking at a free channel here at the collagen and the dermis. And what you see is where the neutrophils swarm, we have depletion of the collagen. It turns out to happen so rapidly, it's not due to metalloproteinases, and high-resolution imaging shows that these collagen strands are being physically pushed away by the swarming neutrophils, and we'll come back to what that means for what's going on here in a short while. So there are these three stages of scouting, amplification, and then the stabilization at the very end. You can't infinitely recruit neutrophils and keep damaging the tissue with their highly inflammatory mediators. And so the question is, what controls the transition between these different stages? So if we look early, the lab had developed a few years ago a means of looking at cell death in vivo using propydium iodide directly injected into the animal to reveal DNA released from dead and dying cells. When you do that, when this recycles, green are the neutrophils, red are the DNA. There are a few dead cells right around the lesion formed by the laser. And you can see the DNA here. Note that it seems to be moving away from the lesion in the opposite direction from the entry of the neutrophils into the system. And again, I'll come back to what that might be telling us in a few moments. And so Tim did a very extensive kinetic study where he knew exactly when the first neutrophil died because he could see when he made the lesion and then when the first cell became propydium iodide positive. And so here are the cells sort of meandering around, and then there's this very rapid transition to highly directional, highly rapid movement, and it always occurred within 1 to 5 minutes of when that first neutrophil died. So it's a correlation. We cannot prove causation here, but all the data tell us that it's the death of this first neutrophil that initiates this rapid recruitment process. Now, how is that mediated on a molecular level? There are a very, very large number of candidates of interesting molecules that may control what I've just shown you. So to set up an internally controlled system that deals with this question I had of what's in the black, variation in the tissue site of imaging, variation in the extent of inflammation, variation in exactly the nature of the lesion, we actually co-transfer wild type and knockout cells in 2 different colors. And then we also do these experiments where we reverse the colors. So we're always looking in the same imaging field, so whatever's in the black is now normalized between the 2 different cell types. It's exactly in the same state of inflammation, and we're not worried that it's the dye label that's giving us the phenotype in the knockouts because we changed the dye label. And when we do that, we get a surprising result. There are lots of molecules you would have thought would be involved in neutrophil function under these conditions that give us no readout. It doesn't mean they're irrelevant. They're surely not uniquely important, but LTB4 and its receptor, LTB4R1, turn out to be especially important. And what we're looking at here is the ratio of mutant and wild type cells accumulating in that swarm. So 1 is equal recruitment. Less than 1 means the mutant cells don't have the same propensity to show up in the swarm. And so if you look at this when it recycles, at the very beginning of the imaging session, these green mutant cells are actually more abundant in the middle. But as the lesion evolves, they're basically excluded from the swarm by the wild-type red cells. And that said, there's probably something going on with cell-to-cell interactions in the actual swarm that parses out which cells can participate. So we looked with a probe for F-actin and could see that where these neutrophils were touching each other, there was constant polymerization of F-actin that says cytoskeletal involvement and suggests that integrins may be involved. And so what we did is to ask if we prevent the integrins from undergoing high-affinity upregulation, which would normally be considered to be important for cell migration as well as adhesion, what does that do to migration into the swarm and participation in the swarm? And we did this by using tail and knockout animals. And when we do that, green are the mutants, red are the wild types, and the phenotype is incredibly dramatic. The mutant cells are completely unable to participate in the central swarm, and if I showed you the collagen part of the image, you'd see that where the collagen is pushed away, there are no green cells. It's only the red cells that are in that zone of exclusion. But the other thing that's obvious is the green cells are migrating perfectly well. And so for those of you who are used to thinking about mesenchymal migration with focal adhesions that involve upregulated integrins, that rule does not apply to myeloid cells. As a PhD student, Tim actually showed that dendritic cells migrate in the complete physical absence of integrins based on quadruple knockouts. Here we only can say something about the high affinity form of the integrin in terms of its function, but there's no measurable difference in any of the interstitial migration behaviors of the neutrophils until they try to participate in the swarm. And so I'm going to summarize what I've shown you and other data in this published work. There is this meandering of cells around the original lesion, and a few of those neutrophils die. We have not been able to characterize yet what the signals are to trigger that death, nor what they release for the next step. And there are many reasons why that's difficult, but we do know what happens after that. There is a damp that's released that induces the neutrophils themselves to make L3T4, the leukotriene. LTB4, sorry. I'm used to talking about CD markers. LTB4, the leukotriene, by those neutrophils. And in a signal relay mode that Carol Parent first described in vitro work, that induces other neutrophils to also make LTB4. So rather than waiting for a diffusive process to spread the signal in the tissue, there's constant cell-to-cell amplification, which accounts for the very rapid transition from meandering behavior to very rapid, very directional migration from many hundreds of microns away from the lesion. Those cells can migrate even without the upregulated integrin affinity that occurs with the signaling, but they cannot participate in the central swarm formation without tightly adhering to each other, and it's MAC1 and LFA1 that mediate that. And why is this swarming important? Why is this tight adhesion important? Well, what's happening is we're isolating the damaged tissue or the infected site from the surrounding healthy tissue or uninfected site. But we don't want to do that beyond the necessary level. So something must constrain the growth of the swarm and the recruitment of the neutrophils, and we suggest that it's these recruited monocytes that make things like Charles Serhan talked about yesterday, the resolvins that counterbalance the pro-attractive forces of the leukotrienes that we've characterized here. And so do we have any evidence for that? Well, I can't tell you that it is the resolvins or it's the monocytes, but I can show you experiments that talk to this issue of whether there's an active process that changes the inflammation to this first step toward the wound healing phenotype. And so that's the stabilization phenotype. And the way we've looked at that is to look at animals that are uniformly red and that use Jennifer Lippincott-Schwartz photoactivatable GFP so that we can mark cells that are already in the lesion by photoconversion. When we do that, late in the lesion generation, we get these types of images where things are very static. Not much is moving. There's a little bit of wiggling. And so there's a problem with this. The most obvious artifactual phenotype in doing this type of intravital imaging is cells don't move. And that's because your animal's been anesthetized for a while and maybe it's not breathing especially well, it's not well oxygenated, or it's getting cold, or you've used so much laser power that you basically fried everything in the field. And so we have to go and ask, is there really an active process that's stopping the neutrophil recruitment, or is it just the end of an experiment? And so to test that, we simply make an additional laser lesion. If everything is effete, that is, the animal's not in good shape, everything is over-illuminated, nothing is going to happen. But if we've achieved a dynamic balance between pro and anti-inflammatory stimuli, we're now rebalancing the system by adding more pro-inflammatory signals. And we ought to now reactivate the system and get migration. And the answer is really quite obvious, that we are not looking at a bad experiment. We're looking at this active process of resolution of an inflammatory event that stops at a level before it causes excess damage, but is still responsive to additional damage. For example, the escape of pathogens from that central swarm, for example. And so recently, Stefan Uderhardt in the lab has been pursuing another model for looking at this other than the ear skin that I've shown you. And there's a particular reason for choosing this, and we're looking at the wall of the peritoneal cavity. And the reason is that this tissue has these embryologically derived fixed tissue macrophages that people know about but have been very poorly studied for what their actual role is in host defense. It also has these recruited peritoneal macrophages that derive from inflammatory monocytes, and it's a site of ready recruitment of neutrophils. So these three main myeloid populations are all present and can be simultaneously studied and imaged in a very accessible manner, though not a simple manner. And it took several months for Stefan to work out how to do this without causing the tissue damage I told you always can interfere with experiments trying to look at the steady state. And so here is a surface picture where the green cells are these embryologically derived fixed tissue macrophages. And what you see is that they survey their local area, but they're not migrating around. If we now add salmonella, which is in red, those little tiny red dots, and we wait a little bit, what you see is this enormous influx of very bright green cells, which are all neutrophils. But if you look carefully, you don't see any of those fixed tissue macrophages move. They're still wiggling, but they don't migrate. And if we look at higher resolution, what you see are the neutrophils coming in and vacuuming up, for example, over here, the surface disposed bacteria, but there's no real uptake by the macrophages. Now, why does this relate to what I just showed you previously? Well, there's going to be an interesting event right over here, which is the death of a neutrophil. And as soon as that happens, what you see is all the other neutrophils start paying attention to that dead neutrophil, independent of whether there's a bacteria there. And the most telling result is to repeat, in a way, the secondary damage experiment I just showed you in the ear. So you've got these tremendous swarms of neutrophils, and now Stefan creates a little laser damage over here. And the phenotype is quite dramatic. All the neutrophils pay attention to the new tissue damage. And so in all the systems we've looked at, cell death and tissue damage is the dominant signal that promotes neutrophil recruitment and swarming. And this, in turn, recruits monocytes and potentially signals to these resident tissue macrophages to mediate the switch from inflammation to wound healing. Now, there's a lot of work to do to look at whether that hypothesis is correct, but it has important implications in cancer. There's a lot of attention paid to myeloid-derived suppressor cells, and they're a major problem in getting immunotherapy to work or your immune response to deal with tumors. Well, wound-healing monocytes and macrophages have most of the properties of MDSC, and it's not unexpected that cell death in tumors will promote the recruitment of such cells, given the data that I've just shown you. And so you don't need to imagine that tumors are doing something very special to protect themselves. They're simply dying and taking advantage, in a way, indirectly, of what the immune system does to deal with tissue damage in recruiting these cells that go to promote a wound-healing environment, IL-4, IL-10, which will then suppress the inflammatory immune responses you need for tumor rejection. So we need to keep this in mind in thinking about what's going on in the tumor environment. Now, innate immunity is great. I've shown you very interesting, very cool behaviors of neutrophils, but it's not completely efficient. We know from the boy in the bubble, if you don't have part of your adaptive system, you don't do well in a non-sterile environment. And so the question is, what limits systemic pathogen dissemination when things get beyond the capacity of the innate system to handle them? And why is this an issue? Well, because there are lymphatics in all of your tissues. And immunologists think of lymphatics as part of the immune system, but people who actually understand the circulatory system realize that evolutionarily, the lymphatics are there to deal with fluid balance. You're constantly transmuting fluid out of your vascular tree, and you need the lymphatics to return that fluid or your volume deplete. But these are large open-ended vessels, and if they were accessible to pathogens that invaded the tissue, then those pathogens could go into the lymphatics, and if you exclude the fact there's a lymph node drawn here, go through directly into the circulation and go systemic. That's not good, and so evolution says, okay, let's evolve lymph nodes and put them in the lymphatics where they can pay attention to those organisms that are trying to hijack the system that way. The problem is when we think about lymph nodes, we think about them as sites of development of adaptive immunity. We don't think about them as primary sites of innate host defense. But the problem is it takes a week or more to develop a very robust adaptive immune response from scratch, and bacteria replicating every 20 or 30 minutes and viruses every 2 to 4 hours. So give your mind a chance to do the computation of what that expansion is over a week, and you realize that it makes no sense for the lymph nodes to only be a site of adaptive immunity, because otherwise they would just turn into food for these organisms, which would then go systemic. And so Wolfgang and Parizad said, okay, there's got to be something else going on here. Let's take a look at what that is. And so to remind you of the architecture, we have these central paracortical T cell zones, these peripheral B cell follicles, but what I haven't told you about is they're myeloid cells, particularly macrophages, that are aligned along the subcapsular sinus space into which lymph drains from the tissue. And if we look under normal conditions where we use a fluorescently labeled Pseudomonas organism with a skin infection, those organisms are trapped in the regions where these macrophages are abundant in the lymph node. But if we use a liposome formulation that will kill phagocytic cells, so specifically these macrophages, now we get robust invasion and replication of the bacteria in the paracortical region, the central regions of the lymph node, and 2 to 3 orders of magnitude more dissemination of those bacteria systemically in the blood or recovered from the spleen. And so the question is, are these macrophages just acting like flypaper, grabbing the particles that come in, or is there really some active process that's constraining bacterial replication and spread in the lymph node? And after a great deal of work, what became obvious, and it's best seen right over here, is that within a few hours of exposure to the skin infection, a whole series of lymphoid cells makes interferon gamma in the draining lymph node. And those are CD8 memory T cells, as well as gamma delta cells, NKT cells, and NK cells. And I'm not going to walk you through all the published data. I'm just going to tell you that the way this works is this preformed IL-18 in the macrophages. It becomes cleaved and released, and it stimulates in a cytokine-dependent cytokine response mode these various lymphoid cells to make that interferon. It doesn't have anything to do with antigen-specific recognition by receptors, and this antibacterial response that's dependent on the macrophages is also dependent on the interferon gamma, so these two are linked. And we can actually see where the interferon gamma is being made by staining here in red. A little bit is over here. And the question is, these lymphoid cells are T cell-like, and you might imagine that therefore they're moving around in the T cell zone in the lymph node. But that means they're not anywhere near these peripheral macrophages that are making these small bursts of IL-8. And we also know from this picture that the cells that are making the interferon are in the periphery of the lymph node, not in the central region. So maybe another alternative is the system has evolved a bit smarter than that, and it's actually positioned these responding cells right next to the macrophages. So again, back to this issue of position and tissue microanatomy, in order to facilitate their recognizing these alarm signals and make the response immediately. And when we do staining and look at where the NKT cells are, the NK cells, the gamma delta cells, and in pictures I'm not showing you, it's more complicated staining, the CD8 memory cells, they all are in the periphery of the lymph node, not in the central T cell zone. And so the headline here is on the top, the lymph node is a major site of innate host defense. And in fact, if you look at large DNA viruses, they have large parts of their genome devoted to evading exactly the mechanism that I'm describing here, so the pox viruses can get through the lymph node and get back to the skin and disseminate. And so we have this peripherally disposed set of sensory macrophages. Nearby, we have these patrolling lymphoid cells that are sensitive to cytokine-induced cytokine production. The inflammasome activation that comes from recognition of the bacteria cleaves the pro-IL-18, gives you the IL-18, gives you the gamma interferon, and then upregulates INOS in these macrophages, so they're better at dealing with intracellular pathogens. And by a pathway I don't have time to describe, there's also IL-1 generated, and it contributes to recruitment of neutrophils into the lymph node. There is actually quite a robust neutrophil response when you infect these animals in the skin, and they deal with extracellular pathogens. So there's a very organized tissue setup here where the positioning plays an important role in making sure the system operates efficiently. And so I want to keep on that theme of efficiency and talk about adaptive immunity. We all know about these cartoons where we activate an immature dendritic cell so that it's good at antigen presentation or co-stimulation or making polarizing cytokines, and then it can activate the adaptive response of CD4 or CD8 T cells, and these CD4 cells can talk to antigen-specific B cells and make antibody, and we're all very happy. The problem with this picture is it's completely fake. And why is that? Because early in an infection, maybe 1 in 10,000 dendritic cells has enough antigen to do anything. And there's only 1 in a million of your CD8 T cells that's specific for the antigen presented by that rare dendritic cell. There's only 1 in a million CD4 cells specific for that, and only 1 in a million B cells specific for that. And so in a lymph node that contains many millions of cells, if you have 1 to 5 of each of these cells, what's the statistical likelihood that they meet up in space and time in the way this cartoon shows? And the answer is, for any 2 cells, very low, and for 3 cells, essentially 0. And efficiency matters here. And what do I mean by that? So you have to think back a couple of million years to evolution, and we have very small mammals. And small mammals need to eat all the time. They have a very high metabolic rate. If you get infected, and the antigen-specific rare cells that I'm talking about here don't find their antigen in one pass through a lymph node, they won't come back for a day or 2 days. Now imagine you're this small mammal, and you feel like, well, you feel poorly. And you can't feed. You die, and you don't pass on your genes. You go out and you try to feed, you can't run away from the predators, you get eaten, you don't pass on your genes. So the answer is evolution says that the immune system has to operate efficiently, and that means these cells must always find their partners essentially in a single pass, in a few hours, in a complex environment. The likelihood that happens by random motility in random positioning is essentially 0. And so over the years, what we figured out is there's fractal biology of lymph node structure and function. As I said in the early introduction, we know that the lymphocytes concentrate in a lymph node and antigen drains to that, so that's putting things together in higher concentration rather than just dispersed in the tissue. If we look within the lymph node, we see that we have a T cell zone. It concentrates those particular cells. There's a stromal compartment within that zone, and we showed a few years ago that the lymphocytes actually migrate on those stromal elements as roadways. And why is that relevant? Because remember, these T cells are looking for antigen-bearing dendritic cells. And where are the dendritic cells? They're sitting on those fibers. So we've now concentrated the T cells and their target cells together under these conditions. And then we showed a number of years later that, in fact, the T cells, when they move, don't only move randomly on the roadway, but if there's an interesting dendritic cell that interacts with at least one lymphocyte, it makes signals, these chemokines, CCL3 and 4, that now bias the movement of these other lymphocytes on the stromal network so that they find this cell 5 to 8 times more frequently than an irrelevant cell the same distance away. And most recently, we've shown that for CD4 help for CD8 cells, a really critical component of adaptive immunity, for example, to tumors, but also to viruses, that this particular subset of XCR1 DC is the focus for this 3-cell interaction. And so there's this hierarchically organized combination of physical and chemical cues that optimize this system to give you good adaptive immunity. It's not a random process. And the design of the system has a lot to do with it operating in an efficient and an effective mode. So let's say it worked and we wound up making effector cells. And I think all the immunologists in this room think of effector cells as dots on flow plots. They're not cells, they're these little dots, and they're 17 positive and 4 negative, or 4 positive and 10 negative, or whatever else you put in your quadrant markers. But they're really cells, and the question is, how do they actually work? Do they make their cytokines continuously or briefly or episodically when they go out into an antigen-containing tissue? What about their dynamics? Do they make their cytokine when they're moving and spread it widely, or do they make it locally and focally when they're stopped? And what happens after they see their antigen? Do they keep responding? Do they desensitize? Do they die? Do they continue to differentiate? Do they divide? There are all sorts of possibilities, but we don't know that from looking at flow plots. And so Jackson and Tetsuya set up a really elegant system to ask that question. We prime animals with a very potent protein, keyhole lipid hemocyanin, and either OVA peptide or ovalbumin protein. And we've put into these animals a limiting number of GFP positive, so trackable T-cell receptor transgenic cells that we know are specific for the ovalbumin. After they're primed, we can come back and challenge in the ear to recruit the cells, and we can do it either with just the KLH, which we've stimulated endogenous cells to respond to and make a nice inflammatory environment, or we can add the ovalbumin in. So we have a very controlled condition to ask what's the effect of antigen recognition on effector cells in the tissue. And when we do that, we see, first of all, perhaps a little surprisingly, we get equal recruitment whether we have the specific antigen present or not. But in the ear without the specific antigen, all the cells are migrating well, but in the ear where antigen's present, many of them have stopped moving, or they're not moving. I shouldn't say stop since I don't know what they were doing before I looked at them. And we can see that quantified here, that in the early phases of the response, most of the T cells that are antigen specific stop in the ear with their antigen. But this can't answer any of the questions that I told you about for a very simple reason. We don't know when the cell came in, we don't know when it first saw its antigen, and therefore we have no kinetic control here. But we can fix that, because now that we know that the KLH challenge brings these trackable cells in, we can do that and now infuse peptide into the animal to make their cognate complexes in an inflammatory environment that has tissue leak and therefore is very susceptible to permeation by these peptides. And when we do that, and now we're going to look at twice the normal speed of the movies I've shown you, and for 14 hours, which is, as far as I know, the longest intravital experiment of this type that's been done. And the cells are moving, and as soon as we put that antigen in, they all stop. And it all, immediately is here within 5 minutes. But then if we let things go, they start moving again. If we add more antigen, many, but not all of them stop. So they're moving, antigen's in, they stop, they're just wiggling, and then some other cells come in and some of them move. And it looks like this, they're moving, they stop with antigen, and over 4 to 8 hours they recover their motility. So what about their cytokine function? Well, it's anti-correlated with their motility. When they come in and are moving, they're not making much cytokine. Within 2 hours of seeing their antigen, they get a cytokine peak. And as they regain motility, they stop making cytokine. We get a monotonic wave of cytokine production under these conditions. And that raises a question. We are doing this within a few hours of administering a large bolus of antigen. Previous studies said peptide MHC class II complexes have lifespans of perhaps days. So why are these cells not stopped anymore, and why are they not making their cytokine anymore if their ligand for stimulation is still present? And one possibility is that in vivo, we don't have a reason to understand this, the antigen is disappearing at a very rapid rate. But more interestingly, and more likely we think, the T cells are tuning to the antigen they're experiencing so they don't sense it anymore. And we'll come back in a minute to why that might be interesting. So we can look at that. We do everything that I just told you about, bring them in unspecifically with KLH, stop them for a period of time with their antigen, let them recover for 6, 8, 9 hours, and now we transfer a new cohort of the same specific T cells that have never seen their antigen in vivo. And they're in a different color so we can track them. And then again, to do the control for what's in the tissue, what's the state of the animal, and so on, we put in a third set of cells that has no specificity for anything in the system, but should migrate around if the inflammatory environment permits it. And when we look, that's exactly what these control cells do. They're moving at the 6 microns per minute. The cells that had been stopped are almost back to 6 microns per minute on average. The new cells we put in stop. So there's plenty of antigen there, but these cells that have seen it already are not paying attention to it anymore. They're not stopped, and they're not making cytokine. So what could that mean? And so we go and read Dan Barber's papers, and we learn about PD-1, something that's very popular these days for cancer immunotherapy. And as his work and the work subsequently in many labs, especially Rafi's lab, shows that within a few hours of TCR engagement, 2 hours here, 3 hours, we get a peak of PD-1 expression on those infiltrating cells. But there are a lot of other negative regulators, so what about those? Well, it turns out CTLA-4 does not go up on these cells. LAG-3 does not go up on these cells. They don't downregulate their T-cell receptor. They don't lose CD28, and they don't even express PD-L1. So pretty much the only candidate from this short list of interesting candidates is PD-1. So the obvious experiment is to block the interaction of PD-1 with its ligand. So we do that. And so here, in this particular experiment, with an isotype control, they're back to full motility in 4 hours. But if we block the interaction of PD-L1 with PD-1, even out here at 6 hours, many of the cells have not returned to full motility. And if we look at their cytokine production, there are 3 times more cells still making interferon gamma when we interfere with that negative regulation. So PD-1 is clearly playing a negative feedback role, and this is essentially a normal inflammatory immune response. It's not a chronically infected animal, it's not a tumor-bearing animal, and it's not a tolerized animal. So you can't think of PD-1 and its function in the context of all these pathological conditions. It actually has a very critical, important role in normal immune homeostasis and regulation. But then the question is, why would you waste effector cells? Why would you turn them off under these conditions? But the answer is T cells have to worry about how much tissue damage they're going to produce, and the only way they can tell whether they should keep making their cytokines is by tracking the level of the pathogen. But they can't do that directly, so they have to measure peptide MHC, their ligand. And so what they're doing is tuning their sensitivity to that ligand. If it goes up, if they're not really effective, they can still work. But if it's constant or going down, they've tuned and they don't do it. But now new cells coming from central memory cells, like the cells we transferred, are able to respond, and they can push the pathogen load down further. So you run this essentially continuous ratchet that relies on new effectors in order for this to work. I'm going to go through this, I think, much more quickly just to say this is a very general technique. It works across many different tissues, as I'm showing you here. But there's a problem. That even though we can look in the liver and in the gut and in the kidney, and we can go and even look in the lung and in the bone marrow, there's a problem. And that is, we look at a very small part of even a small mouse tissue. This is how much of a 1 millimeter mouse lymph node we actually live image. And so there's a lot in the black, and it's not just the black in what we are imaging, it's the black that we don't image. And we don't know what's going on there if we don't look. Well, histochemistry is great for doing that. The problem is that in modern immunology, to identify any particular cell is usually 2 to 4 different markers, and you want to look at a couple of different cell types. You're up to 9 to 14 colors, and most people use red, green, and look for yellow when they're doing histochemistry. So there's a mismatch. So Mike Gerner took on a few years ago to solve this problem and developed a technique called histo-cytometry that lets us do things the equivalent of very highly multiplex flow cytometry in situ, so we can have cell positioning with phenotyping, and I'll show you some of the other tricks that go with this. So we're now up to 10 to 14 colors. It's not a single image. This is what's called a tiled image, where we go across at high resolution and stitch everything together, so we're looking at a very highly resolved image. We deconvolve to even improve it further. We then worry about spillover of fluorescent emission across channels, so we use compensation, like on flow. We begin to gate on the different fluorescent signals we're interested in, but we'd like to assign that fluorescence to cellular objects, so we use software to actually create those cellular objects. But now those objects have fluorescence associated with them. But that's the type of data that you use in flow. So we can now take those data and put them into FlowGel or an equivalent program and begin to gate as if we were doing flow cytometry, but we're doing it with the cells that are in the tissue specimen, where we have also in every cell object the spatial coordinates. So we can then print that back so we know exactly where the cells are. So we know how many there are, and we know where they are, and we know their phenotype. It works with non-human primates and human samples, and I'm going to go very quickly through an example also where we can do antiphosphoprotein to look at signaling, and I showed you anti-cytokine already. So we can look at signaling state, function, phenotype, and location all at one time in high resolution. So we've done this, and Michael's done this for all the different dendritic cell populations in lymph node, and perhaps not unsurprisingly, as I told you, things are not all randomly intermixed. These cells live in very different compartments preferentially within the lymph node. So he's gone on recently to relate the spatial positioning to function. So here we zoom in the subcapsular sinus space where the lymph drains in. Down here you see the capsule of the lymph node and the floor, the lymphatic floor of the subcapsular sinus. And if we zoom in here, we see that there are these green cells, these 11B positive dendritic cells that live embedded in the floor of the subcapsular sinus space. And if we do serial reconstruction of an entire mouse lymph node, I'll let this run for a second, what you'll see is that those 169 macrophages that were involved in innate immune responses live almost exclusively over the dome of the B cell follicles. But in the region of so-called interfollicular space that connects to the T cell zone, it's these 11B dendritic cells. And if we look now live, where this is the capsule, this is the lymph flow, here are the 169 macrophages over the follicle, and here are the dendritic cells, they're both waving processes in the lymph flow where they can interact with particles or antigens that are draining directly from a skin or tissue site. And in fact, if we look at higher resolution, we can actually watch micron-sized particles representing bacteria or vaccine components being captured directly by those dendritic cells. And if we do vaccination experiments where we've eliminated migratory dendritic cells, these cells provide full protection in a vaccine mode against, for example, Pseudomonas infection. So they're really rather critical for the function. Let me end in the last few minutes by pointing out some of the ways we've used this imaging approach to get unexpected answers. And this is work that Jiddu Liu in the lab pioneered. And what he was asking is, over what distance does cytokine signaling work in vivo? How far do cytokines act? Over 1 cell diameter, 10 cell diameters, 100 cell diameters? And then unfortunately, live reporters for the 2P imaging didn't work well, so he started doing staining for p-stats. And when he got to p-stat 5, he got a surprising result. So this is a section of a mouse lymph node. Cyan are the B cell follicles. Dark is the T cell zone. And the green is a reporter for regulatory T cells, marks cells expressing FOXP3. And what you can see is a small subset of those cells are in these very bright p-stat 5 positive clusters, almost always in the periphery of the T cell zone. And we can see that that's actually nuclear, as it should be for activated p-stats. And the surprising result was we get exactly the same result in germ-free animals. So the notion that this is a response to immunity to commensals is not correct. We're actually looking at autospecific cells that are reaching the state of cytokine production on a regular basis in an otherwise intact animal with a full complement of regulatory T cells. We know that the p-stat 5 comes from IL-2 signaling, and we can even identify the one IL-2 producing cell right in the middle of these clusters using this imaging approach. And so these are cells that are getting activated without PAM signals in these germ-free animals. So this notion that you need the adjuvant effect of bacteria to get to cytokine production is also not correct. There is enough co-stimulation, enough antigen, and enough affinity of these cells post-thymically for them to become activated under these conditions. So what about the role of the IL-2 that is being made here? There are data that IL-2 plays an important role in regulatory cell function. So Jiddu tested what happens if you have a wild-type responding cell or a cell that can't make IL-2, and most people would say, well, it won't proliferate. But in fact, IL-2 does different things in vivo than in vitro, and what's clear is that under these limiting conditions, the normal condition is very few T cells interacting with an individual dendritic cell. But if we remove the IL-2 that the Tregs can capture for this pSTAT5 signaling, we get this massive over-response. So eliminating the IL-2 eliminates a negative feedback regulation mediated by the Tregs. And the clustering, I've shown you the T cells stop and they cluster on their presenting cells. So seeing those clusters implied to us there probably was TCR involvement, T cell receptor involvement. So we got together with Sasha Rudensky and showed that if we delete the receptor on mature regulatory cells, which his lab had shown gives you lethal autoimmunity within 4 weeks, we now go from the state in which the pSTAT5 positive cells are all Tregs to these clustered cells where the pSTAT5 positive cells are all the effector cells that are now able to mediate their function in the absence of the co-clustered Tregs. And so to put this together, we have these self-responsive cells that are able to reach a state of IL-2 activation and production in the steady state in a normal animal that does not have overt disease. There's TCR-dependent clustering and increase in suppressive function of Tregs around what are mainly migratory dendritic cells that we can identify by the histo-cytometry. And they have high CD25, so they're able to capture preferentially the IL-2 that's made by these proto-effector T cells. That also further increases their suppressive activity, and that combination plus the IL-2 capture, because IL-2 is important for effector differentiation, now prevents these cells from being harmful to the host. And the critical point is that we normally think of Tregs as keeping the system quiet or turned off, but it doesn't do that. The system is on, but the brake is also on at the same time. And if you take that brake off, now you get explosive autoimmunity because these cells are always reaching activation. And that's a very different picture of how immune regulation works. I'm going to end in the last 1 minute by saying that we're greedy. We've been trying, to look at things live, and we see this little tiny region. I showed you sections. We look at a lot more, but that's still only, say, a 20-micron section. We'd like to look at things in 3 dimensions. The problem is that in most tissues, there's a refractive index mismatch that scatters the light and gives you these kind of opaque-looking tissues. But there are tricks you can use to normalize the refractive index and clarify tissues so they become transparent and you can image at depth. And so this is not a cartoon. This is an image of a mouse lung stained for mostly matrix proteins and collagen from the level of individual alveolar epithelial cells all the way up to the conducting airways in half a lung. But this was an old way of doing the clarification, and we decided this wasn't good enough, and who's we? So we is actually Michael again and Weijie Li, who did an incredible job at redoing the whole idea of how you do this clarification, and has come up with an incredibly simple but also unbelievably effective way to do this. This is now a mouse lymph node that is completely unsectioned, completely intact. It's only stained in 5 colors here. We could do it easily in 10 colors. When I animate it, what you'll see are the fat yellow things are the HEVs, and they have this beautiful branching architecture. Now the B-cell follicles come on. But because we have all the 3D information, we can optically section this lymph node any which way we want. There's no sampling error. We don't have one particular angle that we did a cut through a fixed tissue on. We can look everywhere, which means we don't miss any cells. And so we're now beginning to think about, can we look at a naive response without transgenic cells, because maybe there are 4 or 5 responders, but we can find them and phenotype them in this tissue. The other thing is, as this plays, what you realize is you can see this beautiful architecture in 3D, but if you look at these sections, there's no hint that there's that branching architecture. You never would get it if you did a couple of sections and just looked at those slides. We've also worked at algorithms for segmenting these data sets, so we now can do this, which is take the image, do what I told you about putting things into FlowJo, gate all of those, and then print back all the way through the entire tissue where all the different gated cells live and quantify all of those cell types. And for those of you who are not immunologists, we don't only look in lymph nodes. This is brain from a confetti mouse that has multiple colors and different neurons. And we'll let this zoom in a bit, and you're going to see how good the resolution is. In what is not a thin section, as this rotates, you'll see that it's actually a volume image where we can even see dendritic spines and look at the organization here in 3 dimensions. But the nice thing about this in comparison to other methods is we can also stain. We don't only look at these intrinsic fluorescent proteins. And so here we stained, we meaning Weiji, for glial cells. And we'll let this play through. And this is the second to last slide, so I'm almost finished. And you can see the level of resolution we get in the context of this intrinsic green genetic label, the very fine processes on these astroglial cells that we can stain using this approach. And so we're working on extending this to non-human primate and human samples in 3D and in all these colors. We're trying to see if we can look at specific antigen. That's proved very difficult. And we're desperately trying to incorporate multiplex RNA fish so we can look at transcriptomics at the same time we do phenotyping, function, and signaling. And then with Harry Schroff of NIBIB, we're working on new microscopes that will improve our resolution and the speed of collecting these enormously large data sets. And so I'm going to end by saying I've spent most of the day talking about imaging immunity, but the laboratory of systems biology is not just people doing imaging. We also look at the molecular level, create mathematical models of how signaling works within cells, but also create synthetic cells with real biochemistry that are interacting, and these are cells that are simulating the swarming of neutrophils that even include LFA-ICAM interaction and signaling under those conditions, all in support of what we call a multi-scale approach to integrative immunology. Thanks very much.
So we have time for some questions, if anyone has any questions or comments of Ron. So Ron, I couldn't help but think as you were going through one surprise after the other of the level to which you could push this, what is the limit on this? I mean, when you really think about what you can do, you put up there non-human primate and human samples. How far are we away from doing that? Because there are certain, obviously, things that you want to see in non-human primates that you can't see in a mouse.
Well, so for the human work, we've collaborated with Rick Kaup's group in the VRC, and we can, in the sections, get very nice multicolor staining and do the histo-cytometry. So this is not a question of whether it can be done. It has been done. And we're working out much larger panels now. We're up to 20 to 30 different colors, and we have to then put them into panels of 10 or 12 each to go and do this. There are some other methods that people are proposing that might allow you to do iterative staining rather than all the colors at one time and then go up to 30, 40, 70 parameters if you really wanted to deal with that. The problem becomes the data analysis at that point. And there is a resolution requirement for doing the quantitative work. It's easy to collect pictures. It's harder to collect quantitative data, but this can go quite far.
Question?
Well, congratulations for the pioneering work. So 2 questions. One is, what is happening in the brain versus the rest of the body? Because the cell concentrations and different types of immune cells are different in these 2 categories of tissues.
So Dorian McGavran here at NIH has a very strong program of using these same types of imaging tools looking at the brain. And so if you want details on what can actually be seen and what is seen, I refer you to Dorian's work. I should point out that even though people think of the brain largely as a privileged site, it's not. There are immune responses there. People have recently reported dendritic cells that were missed previously, and Jimmy Carter says he doesn't have any melanoma in his brain anymore after getting PD-1, anti-PD-1 antibodies suggesting the immune system is perfectly active in that environment.
And second question relates to, is nature favoring fighting infection rather than killing tumors?
Tumors are not let me put it this way. There are data that it's important that your immune system prevents the evolution of tumors in organisms, but we tend to think, with some rare unfortunate exceptions, of tumors as consequences of aging, and evolution doesn't care about people who don't reproduce anymore. So it's mainly the infections that are going to get you early, and that's a primary driving force. But it's clear that there is, but let me be absolutely clear about this. What's emerging right now is that in the future, oncologists will all be immuno-oncologists, because there is now pretty much a one-to-one correlation. The tumors that have no good intrinsic immune stimulation and response are the ones that the drugs don't work on because they can't cooperate with the immune system.
Thank you.
My question is sort of related to the last question as well. I was very curious about your comment dealing with the fact that with cancer death, you'll have a neutrophil response, and that will hinder an immune response. And I'm curious is whether or not a patient that has neutrophilia has difficulty in developing an immune response to, let's say, a vaccine.
I can't give you a clear answer on that. I don't have any data, and I don't know anything in the literature on it. I can point out that in the early work we did with David Sachs for Leishmania, the neutrophils come in and take up the organisms. They can't kill L major. Eventually, the neutrophils die. That is a signal to the macrophages and the monocytes that the neutrophils have sort of done their thing, and the system should go again into this kind of wound healing or tissue repair mode, and that actually makes the macrophages a better vehicle for the growth of the L major, and the animals do better if you pre-deplete the neutrophils. So in an analogy to what you're talking about, keeping the neutrophils from recruiting these suppressive myeloid cells, or the ones that put the system into this anti-inflammatory mode, could be beneficial.
Thank you very much.
Yeah, my question is a little bit more practical. In terms, is this your technique, would it be adaptable to surgical pathology in terms of using your technology of looking at metastasis in lymph nodes? They're always a hard time to stage out tumors in terms of looking at metastasis, not necessarily of lymphoid tissue, but of cancer tissue. And in terms of tumor margins, it's always a problem in pathology when you're hitting tumor to see if you've actually got a few extra outlying cells in the margins, and using immunoperoxidase is never really practical, but if you have this down to a cost-effective basis, you might actually solve a couple of practical problems with lymph node metastasis and tumor margins.
So in January, we'll receive our first shipment of human lymph nodes from resections in melanoma and breast cancer patients, some of which have been pathologically typed to be involved or uninvolved, and we will see what we see doing exactly this in human samples under those conditions. So we're trying to do that. We also are under discussion trying to work out how to make this a high throughput technology that can, as you say, become cost effective. So we're actually working on those right now.
Okay, thank you.
Thank you very much, Ron. We have a reception with some light refreshments in the library if you would like to join Ron and all of us to chat a little bit more. Ron, thank you.